Thank you for joining the Blueberry Educational Conference. I'm Federica Rossi. I'm a PhD student at UGA, and today I'm the moderator of this session. I'd like to introduce our first speaker, Massimo Iorizzo. Dr. Massimo Iorizzo from North Carolina University. He's an associate professor, so please, Dr. Iorizzo, the floor is yours.
Okay, thank you, Federica, for the introduction. So, good morning, everybody. I'm going to talk about the VacCAP project, and then I will introduce what the project about and what we have been done in the last 5 years as part of this project. So, before I start, I want to highlight what's the value of this large multi-state, multidisciplinary project. So, in modern breeding, as we start to understand, it's already a few years that we are in the area of modern breeding, a big need for implementing modern breeding. It's development and implementation of genetic and phenomic resources in the breeding program, which allow to implement marker-assisted breeding, genomic phenomic selection, or precision breeding. With that goal, then the goal is this allow to reduce the time taken for developing a new variety, increase the accuracy of the selection, and also increase the number of traits that we can simultaneously select in a breeding program. Now, what does it take to establish a modern breeding program? So first of all, need to be established the resources, including genome, phenotyping that are high throughput and accurate enough, and then cost-effective genotyping platforms. Then we need to understand the genetic of each traits. The hereditability, what is the genetic component controlling the trait, so which we call the genetic study. And then depending on that, then we have couple paths. We can, for traits that are highly heritable and stable across the years, we can use a marker-assisted selection, where for traits that are mid or high heritable but complex genetics controlled by many QTL or many loci in the genome, then we need to use another approach, genomic or phenomic selection, for implementing the breeding tools. And then finally, we move into the implementation, again, marker-assisted selection or genomic phenomic selection. These are somehow the steps that are needed in order to move into the build up modern breeding capacity. Now, all these take a lot of funding, coordination, and then it's very time consuming. It does take time to set up all the steps and the need for implementing these tools. So one of the program funded, supported by the USDA, that has allowed the many crops expanding breeding capacity, modern breeding tool, has been the Specialty Crop Research Initiative, and there are many examples of CAP project funded in several crops. And that's the project about the VacCAP. It's funded through the USDA, through the SREI program. It's a $12 million, over $12 million project that involve over 50 scientists from 25 institutions from US and international collaborators. And the project, the goal of the project is to link DNA to fruit characteristics and fruit quality. In particular, appearance, texture, chemical composition, and in terms of quality, taste, flavor, shelf life, consumer preference, and resistance to bruising. So why fruit quality? Fruit quality because that was the priority in 2019 when we did a survey, 18 when we did a survey with the growers across the US, was the top priority for both blueberry and cranberry, in particular firmness, flavor, and shelf life for blueberry, firmness, size, and anthocyane content for cranberry. And these collectively were over 500 industry representatives that responded to the survey across 14 states. So the project evolved around 5 objectives: establish DNA tool, discover DNA marker linked to fruit characteristics and fruit characteristics linked to fruit quality, deliver DNA marker and advanced selection of progeny with improved quality, and then assess the socioeconomic value of quality characteristics. And then the 51, it's engage the stakeholders and the partners. This is all the PIs that have been involved in the grant. And I'm just going through the objectives and then just summarize what we have been doing since we are at the end of the project. So objective 1 was establish genomic and genotyping resources. In this objective, we have developed 7 new genomes. We developed the Vaccinium pangenome and 5 linkage maps. What we did with this data, we basically used the data and we found that all the Vaccinium genomes are highly collinear. There is high collinearity between, for example, Vaccinium darwii and Blueberry, Myrtillus and Blueberry, but even cranberry and Blueberry are highly collinear. We demonstrated the tetraploid Blueberry behavior of that is an autopolyploid. And then we started to use the genome to discover genes that are controlling some of the traits that are important for blueberry or cranberry. Here it's an example of discovering the gene transcription factor controlling anthocyane accumulation in Myrtillus and in cranberry. Then we developed 2 genotyping platforms, one for blueberry and one for cranberry, that are able to screen between 22,000 to 17,000 markers at a much reduced cost. And then to lower the price and make this platform available, we created a consortium, a genotyping consortium, the Vaccinium Genotyping Consortium. And the consortium has 15 members, has been be able to bring over 28 projects, 30 studies, and screen over 16,000 samples. And we have already published the data for blueberry, and we are working on the cranberry paper. And the platform has been used in the project for carrying a lot of several genetic studies. So objective 2 was more about discover marker and fruit characteristics that maximize quality. One component was to develop accurate and high throughput phenotyping method, then linking the phenotype to the quality in terms of shelf life, bruising, and sensory, and then discover QTL, or region of the genome, can control the fruit characteristics. We measured over 90 parameters, 80 chemicals. So what we learned through this process, this objective. So for blueberry, we have been developing a new method for evaluating sugars using near organic acid and volatile and cell wall composition, texture, appearance and structure, and fruit cluster, and then we develop a barcode system for tracking samples. We have been evaluating over 3,700 samples for 2-3 years and conducted over 30 studies. Some of the information we learned when we talk about linking fruit characteristics to fruit quality. So we have identified some mechanical texture parameter measured with the texture analyzer that can enable a breeder to select for post-storage, for reduced bruising ratio, and for increasing higher firmness, but sensorial firmness. We found that larger size, it's related to less water loss and wrinkle. A total sugar to acid ratio lower than 20, it's usually associated with increased willingness to pay. And we found a threshold of organic acid below 80 milligram per gram of dry weight as a sort of threshold for don't have reduction of willingness to pay by consumers. So those parameters are parameters that benchmark that the breeding program can start to use for improving quality. In terms of genetics, we discovered over 400 QTLs for blueberry. I'm not summarizing the information for cranberry here. But then among these QTL, the one in green here are QTL that we found to be stable across the years and a major effect that we are considering now for marker-assisted selection, where the one highlighted in red are, for example, size, sugar, texture, are more complex traits that should be considered for genomic or phenomic selection. So objective 3 was more about delivering molecular markers and use the data for making selection. So the first step when we want to develop a molecular marker is to identify, validate the QTL across the germplasm, then develop the DNA assay, and then use the information for selection. So we identified stable QTL. These are examples of QTL identified across different germplasm. For example, quinic acid was identified, the same QTL in this chromosome 4 was identified in 3 different studies. The same for shikimic acid, 2 studies for citric acid, 2 studies for eucalyptol, chlorogenic acid. So this is a sign that these QTL are stable and potentially can be used for developing marker-assisted selection for these traits. Then the program at UF, which will talk, will be the speaker, Dr. Munoz after me, started to use some of this information to develop a DNA marker for eucalyptol for marker-assisted selection, and eucalyptol is negatively correlated with consumer liking blueberry. Through this process, he was able to reduce the selection or eliminate at the seedling stage over 6,000 plants, 50%. Of the seedlings. Then we were able to use the information also to make selection in the breeding program. Here are some examples of blueberry selection in the Oregon breeding program that had firm, good flavor, crisp or long shelf life, large size, and then for cranberry as well, you can see these selections that were very large and then also with different color. So objective 4 was more about socioeconomics, link the quality with the value, economic value, and I'm not going to present any data on this. Later today there will be a talk by Dr. Canales, collaborator on the project, that will present some of the data. The objective 5 is the extension, engaging the Vaccinium breeder and stakeholder groups through multiple activity, developing website, newsletter, presentation, publication, webinar and video, workshop and annual meetings. And these are overall outcomes. So basically all the inputs from the other objective are used as an input for the extension activity. We have maintained and developed 2 websites, 2 social media, 22 newsletter, news article, over 200 presentations, 64 publications, 18 workshop and video, and workshops and webinar, 11 video, and then 4 annual meetings with reports delivered to the stakeholders. That's also available on the website. Overall, this is the GDV, the database for vaccine species that has been maintained through the VacCAP project. And then the VacCAP project contributed to over 80% of all the genomic resources, genomes present in the database, over 58% of the genetic markers and 24% of the QTL. And keep in mind that some of the study are still to be completed, so we are working on these numbers are just going to increase. The newsletter, we develop and release, distribute a newsletter on both the GDV newsletter, that is more dedicated to genomic resources, but also to the extension website. And you can look up the last 2 issues of the newsletter where we have started to summarize the impact of the project for each objective. We released the objective 4 socioeconomic newsletter about summarizing all the findings of the objective 4, the socioeconomic, and then the genomic and phenotyping, the objective 1. You can subscribe to the newsletter, and Then what is the impact of the project? Has the project resulted in some change, shift in the community? So it has expanded use of DNA tools in Blueberry and Cranberry, and as a result, what we noted is that breeding program, private and especially the private industry are starting to develop genetic stock that are more adapted to perform a genetic study and do genomic selection. We expanded capacity to discover and characterize genes. So research now is shifting toward functional characterization of genes rather than just identifying a molecular marker associated with genes. Some breeding program initiated to adopt DNA marker in the program or new phenotyping methods, which promise more effective way to select new improved cultivar. Advanced the selection of new cultivars, established a solid interdisciplinary framework to link fruit quality and fruit characteristics, and expanded collaboration across the vaccine community, which limit duplication and increase outputs. So when we go back to the scheme that I presented before about what does it take to establish a modern breeding program, where we are with after this project, I could say we can start to say that we can put a check mark, at least for now, on developing genomes, developing phenotyping for fruit characteristics, and cost-effective genotyping. We started to understand the genetics of the fruit characteristics, and we start understanding which direction to go for those fruit characteristics, either through marker-assisted selection or through phenomic selection. And then we initiated efforts to develop a molecular marker for one of the traits that we were evaluating. So where we are going now? The project is about to end, and then we are planning a new grant. So then what we did last winter, we went back to the industry and we asked again, what are the priority now? So, and there was some change in the priority selected by the industry. We collected about 370 respondents, and fruit quality remained a top priority for blueberry. And then mechanical harvestability and disease resistance come soon after fruit quality. The new project will be heavily focusing on characteristics, developing a method for evaluating characteristics related to disease resistance, mechanical harvest, and yield, assess the genetics of this trait, and then for the fruit characteristics we worked on the VacCAP, the first project, we will move forward this trait into more the development and implementation of the tools in the breeding program through marker-assisted selection and genomic selection, and also evaluate G by E effect. This is an outline of the new proposal. We'll still have the 5 objectives, and then we have submitted the pre-application that has been supported by over 64 partners, including industry and academic representatives. Again, I want to thank you, the VacCAP team, all the people involved in the project, and the support of all the industry organizations that have been supporting the project. And thanks to the USDA for the funding. And thank you for your attention. I will be happy to take any question.
Those studies are two varieties, Randall and Bush?
No, yeah, the focus was largely on northern and southern high bush. Those are large grants, but still, involvement of several groups take then to focus on some major area, and then we had to decide, for budget reason, we had to decide to focus on the main market, which is northern and southern high bush. Yep. The other complexity is the rhabdi require some tweak on the genotypics, the models to develop, because it's hexaploid. It's a little bit different than the tetraploid. Yeah, no, we didn't look at we didn't evaluate the effect of the temperature. We know that there is an effect, but I have to say we did 3 studies where we used a different temperature, independent study, and then we looked at what are the mechanical texture component that changed during the storage. And it was interesting that across the 3 studies were exactly the same component, texture component that were changed. We do know that, for example, in Florida, they had to use 1 Celsius for storage for the study, and we noted a lower water loss. Compared to Oregon, where we stored the fruit at 4 degrees, the water loss was higher. So we know that temperature has some change, but overall, the texture changed in the same way across the different studies. Thank you. Thank you. Yes.
Thank you again, Dr. Massimo Iorizzo. And now we can continue with the other speaker, the next speaker, Dr. Patricio Munoz. Is an associate professor at University of Florida. So please, the floor is yours.
Well, thank you, everybody, and for inviting me once more to share the updates about our program. So regarding the question about post-harvest, it's not my topic, but I know that Dr. Steve Sargent, years ago did all the work for temperature effects in post-harvest. So I think if you want to get some leads about that research, it was done by Steve Sargent from University of Florida, among others also as well. So thanks for inviting me to present some updates about the work of the university. Today, the outline of the project of the presentation will be, I'll talk to you about the goals of the program, because if I want to give you some updates, I want you to understand what do we do in the program regarding breeding, extension, research, and training, and then the future of what we expect, right? So one of the great things about our industry right now is that the consumption of blueberries keep increasing, and that's why it keeps us all busy, right? And that's why we keep doing what we are doing. Now, in our program, what are our goals? So we have 4 major goals in the breeding program at the University of Florida. One of them is the extension, breeding plus extension. And the other part, it will be research and teaching. The first section is more applied, right? And it deals directly usually with growers, because we release cultivars and then also we follow up on them and try to help them. So basically developing varieties and then support our stakeholders. While the second part, the research and teachings are more, more basic, or they are less interaction usually with the growers. So I always like to start, to give context regarding the, the birth of the southern high bush blueberries and where is it coming from and where we are nowadays, right, regarding the program. So the domestication, 1911. The University of Florida started working in 1950s in developing the new varietal type that nowadays we call Southern High Bush, right? And since the first variety that hit, it took around 26 years, Sharp Blue, for the first variety to be released. Just anecdotal, Sharp Blue is the mother of Biloxi, a very successful variety as well. So since that moment, a new varietal type is established and the program started releasing many varieties. The work of Ralph Sharp, he was the visionary that, thought about making this happen, right? And then Paul Larine, as you might know, he was one of the breeders that materialized this vision from Ralph Sharp. So I'm the fourth into this, the breeders at the University of Florida. And since I have been in the program, we have established around 13 new varieties for our stakeholders. Recently, I gave a talk to the Florida Blueberry Growers Association, but I know that here in Georgia also they use key crease the varieties, so I just wanted to highlight a couple of the varieties that you guys use here. And at the same time, for example, Fardin, high yield, we call it reliable mid-late yield. But also, just mention very briefly, and I'm not going to cover the numbers because this is the reason that we create the phone app for you guys to look at the numbers directly. But we have developed the variety Sentinel with high yield and good flavor. Also, we have developed a pink variety recently that is being licensed globally, or the variety Albus that I know that some of these varieties are recently being established here in Georgia as well as in Florida. And just last year, we released a couple of new varieties, and probably I'm going to stop here to speak a little bit more about these ones. The goals of the breeding program, for all producers, we have many stakeholders, so we have the final consumer that in reality is only looking for experience. But we have the grower, on the other hand, that is actually looking to be profitable with the product that's developing. So we have to balance these goals, and at the same time you have probably the nurseries that is looking to have high, propagation capacity on the plants, right? And then the cheaper or the marketer that is looking for having high firmness of the fruit so then they can move the fruit efficiently from one point to another. So in the program, we have to balance all these goals and all these requirements by your different stakeholders. Now, yield is always going to be the number one because without yield is the one that pulls the first part of the chain, right? So if we don't have any yield, the producers is not able to make any money, and then basically we don't have an industry. So, but more and more important, we have been paying more attention to the consumer, because on the other hand, if nobody's consumer the blueberries that we are producing, we're not going to have an industry in the future. So as I mentioned at the beginning, we are increasing the consumption of blueberries, but it's very important that we keep increasing that consumption because the volume of blueberries that are getting produced globally is getting quickly faster than the capacity of the people to eat these blueberries. So anyway, Falcon is a new variety that we released, and then the characteristics is that jumbo berry, very firm, with high yield and very good flavor. And actually, this is a true photo of some primocane fruit. A couple of years ago, I got that in Waldo, Florida. So I'm just going to stop to give you an idea of the way that we work. We put variety trials across the whole state. And then we keep track of this yield for several years until we decide to release a variety. In this case, what you see the location, you see the year where it was established, the planting. And in orange, you see the peak, the beginning of the season, the peak, and the ending of the harvest. And then in the numbers that you see in the box is the yield in pounds per acre. And then the density of planting, while in some cases might vary a little bit, is around 1452 plants per acre, with some exceptions, like, for example, this one in the tunnel. So very high yields documented. We are going to stop at this point at the university to document the yield of this variety because now we know that it's a high yield and then it's good for our stakeholders. A second variety that we released last year, we call it FL 19006. And people keep asking me what is going to be the name. The name is 19006. So maybe we can all agree that, the name doesn't matter as much because once people learn the name and then whenever we start talking about this one, we introduce it as 1906. And now everybody knows it with that name. So we don't need to change that. So also it's a very large and jumbo berry, firm yield, aroma and crunching. One of the way or another, this is one of the varieties I really like that finally we are being able to somehow check all the boxes that we are looking for. Very high yields also documented, and also we're going to stop tracking the yield for this variety because we have all the yield and fruit quality data that we need at this point. If you need more information about these varieties or any of the other ones that we have released, all the information is in the phone application that we have developed that I'm going to be introducing in a moment. So the second thing that I want to do is just to give you an idea of what is the impact and what is happening because we keep releasing these varieties and then, or for example, the information that Massey was presenting regarding all the use of technology that I'm going to cover in a second as well. Where is that coming? So it's coming in in the form of new varieties that our producers or stakeholders are able to use and actually are creating an impact. And sometimes the research that we do seems a little bit disconnected to what you guys are doing. But believe me, it takes some time, but it gets there. So for the people from Florida, you know, I am sorry that I'm presenting some of the slides that you already seen recently, but this is the percentage acres by cultivar currently. So with the help of Doug Phillips, which is our extension specialist that is sitting down there, but I'm going to be talking about him in a second as well. He was able to carry a survey, and we still, as you see, we have some areas that we still need to identify some of the area in the north part of the state. But actually we see great adoption of new cultivars in the state, and with that we see an increase in quality and increase in yields. So we divide the state in 3 areas, so north and central and south, and then you can see the case for the central Florida and the case for South Florida as well. So why is this important? It's important because in order for us to make good decisions about what do we need to change or what do we need to improve, we need to know what do we have out there and what is more urgently to be replaced, right? So that's why we carry all this survey and we look at all this information in order to make decisions about what do we need to be working on. The second objective of the program then, or the goal, is to support the stakeholders to maintain competitiveness. And then we developed, a few years ago already, the phone app application that is free for everybody to download. And then you can search it for UF5 Blueberry Growers Guide app. And also is, since the beginning, it was translated in Spanish because we know that we have some managers that need the information in Spanish. So these applications have several modules. And then actually this is an old picture because we also have the chemical module here, which is tied to the government database. Every time that they change anything, it changes automatically in the app. So about what are the products that are registered for blueberries, and then basically in the scouting guide, once you find a problem with a disease, pest, or whatever problem you have, it's also linked to the chemical that you probably need to use. These are just recommendations. We always, sorry, these are not recommendations. These are just for you to know. Still, the recommendation is that you seek to your blueberry specialist in your state, and then obviously send samples and then get a formal evaluation of the problem that you might have. The second thing that we do in this area, and then extension, is the coordinator, and there is Doug Phillips. A lot of what we do regarding direct interaction with the growers is due to the tireless work of Doug that is in the road all the time, I know how many times I have to change tires in that truck, right, Doug? And then basically, he helped us to maintain a direct line with the stakeholders across the whole state. The third objective of the program, as I mentioned, and these are the ones that usually seems more disconnected to the growers or producers, but in reality are the ones that we use to generate impact with the varieties that we are developing. And then in this sense, we have been releasing varieties continuously, but behind the scene, obviously, we have a pipeline to develop products, which are the varieties or the cultivars, but at the same time, to improve the population or the germoplast, so then we can do better in the future. In that process, we use many tools, and then we have different strategies. So many tools, for example, the genomic selection that we have been using, or genomic prediction that we have been using already for 5 years in the program. We use metabolomics that actually help us to predict flavor of many varieties simultaneously, so we don't need to be testing all of them, or doing tasting panels that are very expensive and low throughput. And now we are implementing phenomics, so that actually with one single picture or some images, we can get a lot more of the characteristics of the plants and use them actually to make predictions as well. And then still we use tasting panels for the final products to validate the final products that we have in the program. In research, we are organizing these 4 different branches. So we have statistical genetics. It has to do all with the prediction of the program. Computational biology, that you might think it has to do all with the bioinformatics. Cellular molecular biology, that if you might, has to do with all the validation of genes, what do they do, and also with the gene editing, and then micropropagation or tissue culture. And this area that is becoming more and more relevant nowadays, which is the automation, computer vision, robotics, and all that. Some of the traits that I want to highlight that they are very important. As I mentioned before, yield is the number one. The second most important trait program in the program is going to be size, firmness of the fruit, and flavor. Those are the ones that we push very hard. And everything else, we still push it, but we don't push as hard as those ones. Why? Because with these four main ones that I just mentioned, we can have a very good impact across all our stakeholders. Texture is very important, as Massimo mentioned. With the help of VacCAP, they develop, or we develop all together, methodologies to measure texture more efficiently, because texture is not just the firmness of the fruit. And with these new methodologies, we have been using them now in the program. We have been developed, more methodologies in these areas, and nowadays we have methodologies to screen for different textures for the program. And then hopefully in the future we can have different textures for depending on the product that we're going to be needing. Pollination, and I tell to my entomologists or my colleagues or friends that they are working in pollination or working in entomology, it's like, I'm sorry, but I don't care much about pollination. I care about fruit set. I want that fruit in the bush, right? So it's like, I don't want to see just flowers, right? Because we all have seen that. Right, Gerard? Gerard is laughing here. When we see these blueberries full of flowers, and then when we go to get berries, there is not that many. And we have some issues with, for a while, with different varieties of pollination of fruit set in reality, right? But fruit set is one of the traits that usually gets overlooked because of it's so early in the season, right? So whenever we are evaluating varieties, we are looking at the yield, we're looking at the number of berries that they have, or the kilos or the pounds that they produce. But in reality, yield starts a lot earlier, start with fruit set. So we have been doing a lot of research in this area, and nowadays, some of the research that Dr. Juliana Cromi or Ryan Cullen and other of my students developed is being implemented actually in the process of the pipeline of selections in the program. So to try to avoid, we have discovered, for example, what are the different characteristics that we need to select for flowers for the bees to be able to pollinate those bushes, and then for the producers to have the berries. Blueberry is the foremost expensive, crop for pollination in the country. So obviously, it's one of the traits that is very important to work on. Now, we are not working in this trade because of decreasing costs, a discussion that I was having with some stakeholders earlier today. We are working in this trade because it increases yield. And that's a lot more impacting than actually reducing the cost in this case, for example. We are working, and then we have also developed tools, new tools for selecting using either marker-assisted selection or using genomic prediction for these three major diseases. All in shade here, you can see all the other diseases that we have not developed tools yet, but we are in the process. And we just started another project with Phil Harmon to develop some new tools also for leaf rust that is very important for an evergreen production system in the central and south part of the state. All this was the work of Dr. Lucian Gimire, that recently graduated of the program. And then we have a couple of students that are continuing working in diseases, which is another way that you can see, yield, right? So that's preventing the yield to materialize. Post-harvest, recently we finished, the evaluation of 600 varieties for post-harvest with the idea to learn and develop tools for us to be more efficient at developing varieties that can do, they can have better post-harvest characteristics. And we're taking post-harvest characteristics that go between, all the way to 7 weeks. I heard recently, a great story from a colleague, actually was a student of mine that is working in Oregon now, and he was telling me how by saving fruit, they were able to achieve, higher prices later in the season. And that can only happen if the fruit that you have it has good post-harvest characteristic. Something that in the past, if you think about the time that it takes to consume the fruit here in the country, especially in Florida, we have documented between 3 to 12 days. Basically, the fruit is gone. However, if in any second any moment we need, to save the fruit, they need to have the characteristic for post-harvest, which are different than just firmness of the fruit. Nowadays, we do have tools to do this as well. That's one of my students that put super blueberries there. Flavor, as I mentioned before, is one of the very important traits that we're going to keep working on, and now, developing a specialized flavor or very unique flavors is our next target in the program, and hopefully you're going to start seeing more of that. Now, we have been working in an omic way, and if you don't understand what I'm trying to say, it's basically very general to try to just improve flavor. But I think nowadays the program is mature enough that we could potentially start pushing different flavors depending on what the market is looking for. In that regard, we just published recently another publication regarding what are the thresholds that we need to have for the consumers to like or dislike the fruit, And then, for example, just to give you an idea, we know that when the blueberries, they need to have certain level of acidity, but whenever it goes too high or too low, the consumers dislike that fruit. And with this information, we can use it to be more accurate in the way that we are selecting the products for becoming varieties. Very quickly, in training, we are working in all these different areas already and supporting students and postdocs in different areas. And in the future, what is going to be changing? So we have already implemented, since the moment that I started, the goal was implementing more advanced tools in the program. So we are already using genomic marker assistant selection. We are using metabolomics. We are in the process of establishing phenomics. And then the goals for the futures in that area of research is to implement gene editing that I have, as soon as the year started this year, we have great news regarding that in the lab. Robotics is the next, we know that machine harvest is very important. I think we are there. And then now the next step, what is the future for the program, is going to be robotics. Enviromics is a way for us to predict where the varieties are going to be performing much better. It's a combination of the weather characteristic or the climate plus the genetics, and then so with that we can identify where to deliver the varieties that we are producing. And then functionality in research is something that I would love to see in the industry. And by what I mean by that, I was recently in one of these large trade shows in Europe, and then in one of the person presenting there, they have 15 different types of tomatoes. 15 different types, different colors, different shapes, different uses. That's functionality. And then nowadays, in the case of blueberries, maybe we have 2 or 3 categories. So in order for the industry to be more and then to achieve, the goals that we have regarding consumption, we need to, in my view, we need to hit functionality. We need to develop products for different uses. And I think nowadays, with all the knowledge that we as a community, we have been built, we are at the tipping point that actually we are able to achieve that. So independent of that, blueberries is what it is because of the health halo, and the healthy characteristics. So I say that still we need to keep pushing this message to the consumer, eat blueberries because you can be healthier, right? And with that, obviously acknowledge and then thanks our collaborators across the state that help us to do the work that we do. And then thank you for your attention, and I'll be happy to entertain any questions that you might have. Thanks. Right there.
I noticed the pH is 3.4, so that's a new thing.
So this is the That's the tipping point. So that means that, you know, in one point, if you look at the consumption acceptability of that blueberry with those characteristics start increasing until it gets to a tipping point. pH is a very, let's say, very new. We have always used treatable acidity in the industry. So we are trying to switch into pH because pH is a lot faster to measure, a lot cheaper, and a lot higher throughput. However, honestly, we are not quite there yet. But with treatable acidity, you see that actually the blueberry needs to have certain level of acidity, otherwise it's too plain, you know, and too flat. And then also some flavor characteristics, you are not able to see them. You're not able to sense them, sorry, if the blueberry doesn't have acidity. So also you have different markets, and especially, you know, here in the country, the consumers is used to have certain acidity in the blueberries. But yes, we are getting there. Yes. Okay, I always get this question. So actually, most of the data that you see in those tables, it comes from evergreen, right? And in evergreen, I mean, chill doesn't matter really. So when you go evergreen, I mean, there is no chill requirement. So a lot of the varieties that we have been developing in Central and South Florida, because we had a need, you know, to have better varieties in that area, you know, we have not much information on how they're going to be performing in the north, or for example, in Georgia, or South Carolina, because we are not been tracking, you know, what is the chill requirement of those varieties. So the trialing needs to be done. We have been focused. So as I mentioned, you know, we have north, central, and south, and then we target each area with a specific cultivar. So, for example, I can tell you that the goal of the program is for the next varieties to be delivered in the north part of the country, of the state, and then usually those varieties also perform also more in the north of Florida as well. They work well, but they tend to evergreen in that area because, you know, just to give you context, you know, around Gainesville, we used to have 350 chill hours, the historic records say. Now we are lucky when we get, 150 maybe? So then if you think about that, and then if you don't have a lot of freezes event, the plants tend to evergreen. And because of that, you know, we're getting old. So we are, that area was an amazing area in the past to grow blueberries. Now it's in a transition. So we need to develop varieties that are more adapted for this transition area. Now, if you move into the south, they do amazing. If you move into the north, we don't know. You know, so that's where we are right now. So somebody else? Yes.
Maybe the blueberry is getting bigger and bigger, and if you go to heavier, do you see the future of mechanical harvesting? Will it still be shaping and packing, or relatively by making wrong, or do you see another development?
So let me, in my view, I wish that we were not pushing for just jumbo or super large blueberries because they are in detriment of other characteristics, as I was discussing with somebody this morning as well. Every time that we push too much in one of the characteristics is in detriment of others, right? These larger blueberries, and also, I mean, personally, and this is something that I got some people was not very happy that I posted the other day in LinkedIn about this. It's like they look ugly in the clamshell whenever you have, you know, only a few berries. And then as a consumer, I feel that I'm being cheated, you know, because the clamshell looks empty. You know what I mean? And especially the super large ones. Anyway, so historically, we have been seeing that people relate larger blueberries with softer blueberries, right? But this is because, you know, in the past, there was just a handful of varieties. And then in this handful of varieties, there were some that they were large and they were soft. But actually, after we have studied thousands of different varieties, we know that there is no correlation between these two traits. It just happens that in the past, they selected some large and soft. We have very large blueberries that can be very firm. Firmness is going to be the number one characteristic that is more important for machine harvest, right? And the second one probably I will say is concentration, something that we have been working with Gerardo Núñez that is going to be talking later. Because, obviously you need the yield, but you need the berry to be large, but then you need to be concentrated because there's no point to, be saving money if you have to run the machine harvest every other day, right? So there is, now, machine harvest still is going to be here for a while. Again, as a breeder, I need to be working in the future. When I mention robotics and all these other things, because, okay, that's the way I see where the industry is going, and we need to develop now varieties, in my view, that eventually is going to be the next set of varieties that are going to be harvested by robots, right? And those have different characteristics that the machine harvest variety has. Or it has different characteristics that the variety that is harvested by hand, So, yes, a large variety can be harvested with machine without problems. There is other characteristics, as I mentioned, that they are important. I don't know if I answered your question. I think I went around and I confused myself as well. So, Silfina. So I was explaining to somebody this morning. So breeding works like this. If you make a cross and then you have, 1,000 progeny or individuals out of that cross, kids, right? And then you say, I want them to yield more than, I want them to yield 10,000 pounds per acre. And then you start looking, okay, you say, okay, I have 100 that are able to achieve that, right? And now you say, okay, I want them to be 18 plus. And now I have 20 only that are able to achieve those two requirements. And now you say, wait, I want them to be 13 in bricks. And now you have 5. Right, Sushan? And then you say, okay, but now I want them to be disease resistant. Oh, and now you end up with 2. And then you say, oh, wait, I want them to be early. And now you have none. Right? So the point is that every time that you push too much on one of these traits is in detriment of others. That's the way breeding works. And that's why we need to be balancing all these requirements from the different stakeholders that we have. And also for the different, what the biology of the plants allow us to do. So some of the things that some people without the knowledge wants to achieve is unrealistic. And we do what we can with what we have. So. Yes.
Can you explain how firmness and crispness relate in terms of machine possibility or,
Yep, sure. So easy way to see this, every crunchiness and crispiness are two different traits from the sensory point of view. Crunchiness is the sound that your mouth makes whenever you chew something with your molars, and eyes is the perfect example that is used for that. While crispiness is the sound that your front teeth make when they bite something, and the example for that is potato chips, right? So two different characteristics. However, in the blueberry industry, we use this as one, and we always refer to fruit crunchiness, which is fine, right, for what we need. Every variety that is crunchy is usually firm, and so it can be machine harvested, right? Every variety is firm is not necessarily crunch, right? And so there is certain levels of thresholds in the firmness that you need actually to be able to machine harvest a variety. And that's probably the most important trait, if I understand well, David, what you are looking for. Okay, I need to get kicked out of here. Thank you for your attention. Thank you so much.
And now is the time of Dr. Sushan Ru from University of Auburn. She's an associate professor, so please, the floor is yours.
Thanks for the introduction, Federica, and morning, everyone. My name is Sushan Ru. I am a blueberry breeder at Auburn University. It's a pleasure to come here again to present you some recent updates from our breeding program. So in this presentation, I'm going to focus on some new results from frost tolerance trials that we have in Alabama. And also I will spend a few minutes on a new smartphone app for yield prediction. Dr. Kroor might have some information on falcon in northern region. So it's really nice to have Patricio to start off with very exciting new varieties that are released from Florida. And so we also did some evaluation on new varieties from different programs. And so I'd like to share some information on that. So raise your hand, can you show me if you have overhead irrigation on your farm? Yeah, I see a good amount of people have that. And how about if you run your farm without overhead irrigation, can you raise your hands? Not many, that's good to know. So the blueberry industry in Alabama is a little different from Georgia and Florida. We currently have a bit over 1,000 acres of blueberries and much smaller industry. Most of those growers are targeting the retail market. So currently, Rabbit is still the dominant type growing in Alabama because they're tougher, easier to produce. However, growers are interested in adopting some new southern highbush cultivars for improved fruit quality. So the breeding program at Auburn University was established in 2021, a bit over 3 years ago. Our goal is to introduce and develop new cultivars for Alabama and potentially benefit nearby regions as well. So different from established programs, as a new program, we don't have our own releases yet, but we were focusing also on evaluating existing cultivars from other breeding programs at this point. So for our breeding effort, we focus on both southern highbush and rabbit to meet the unique industry demands. For southern highbush, we want to improve them for stress tolerance, especially better spring frost tolerance. And for rabbit, we focus on early maturity and improved fruit quality. So growers in Alabama have been trying to grow southern highbush, but in general, the success has been quite limited. A major challenge for us is spring frost. Since Alabama is a bit north to Florida, compared to Florida and southern Georgia, we experienced a bit colder winter and more frequent frost events. So throughout Alabama, we have frequent frost all the way till mid-March, and sometimes it carries over till end of March. So different from Georgia and Florida, most of our growers do not have overhead irrigation for frost protection. So one reason is that they simply don't have enough money to install the system, or if they do, most of the farms don't even have enough water to support the system. So our growers have to rely on frost-tolerant cultivars. So in the past few years, the Alabama industry has suffered a lot from spring frost. In 2021, there was a late frost in central Alabama that reduced crop yield by 40%. 2022, I believe a lot of you are still remember that spring frost, and it hit many parts of Georgia and some parts of Alabama and reduced yield by 50%. 2023, it was a really, really bad year for Alabama, and we had a very warm winter to start with, and a late spring frost happened in the end of March that wiped out the entire crop for that year. 2024, finally, we had a good year with no late frost, and as growers are embracing the record high yield, the market crashed. And because of the abundant supplies of blueberries from all over the southeast region, and a lot of growers left their berries unharvested. And as a result, many growers suffered greatly financially. Some of them even decided to leave the industry. So the Wayne County Blueberry Growers Association is the major co-op in the border of South Alabama and Mississippi. At its peak, there were 26 growers in that co-op, and after last year, only 9 members are left. So that's how desperate the situation is, and our growers are really needing more climate resilient cultivars and also some different harvesting window to diversify the market. So a lot of us know that as the flower buds develop from tight bud to full blooms and eventually develop berries, they become more sensitive to cold temperatures. So an easy way to reduce frost damage is to postpone the development of flowers. So we were thinking, is it a good strategy to introduce late blooming southern hybrid cultivars so that we can better mitigate frost damages and while diversifying the market? In order to test that idea, we worked closely with breeders from all over the Southeast to introduce the newest selections and cultivars released or developed by the breeding program at North Carolina, Georgia, Florida, and Mississippi. And so we're testing over 50 collections in Central and South Alabama. And so if we look at historical weather data on chilling hours, and Central Alabama is little similar to Castle Hen in North Carolina, but with slightly less chilling hours. And if we look at South Alabama, and it's very similar to Alma, Georgia, so but with a bit higher chilling hours. So in general, our harvesting window is usually a week or 2 later than Alma, Georgia. So it's quite comparable. And so the idea is we're hoping that based on the similarity of chilling hours and temperature, we're hoping to have some North Carolina cultivars or varieties work for Central Alabama, and maybe some Florida and Georgia and Mississippi varieties work better for South Alabama. And is that the case? We'll find out. So this trial has been going on for a bit over 2 years, and the season of 2023 was the first season that we were able to collect some data on North Carolina and Georgia cultivars. So that season started with very warm temperatures, and which induced the flowering of some of the cultivars, and some of them even started blooming before November. And then we had colder weather in January and several cold spells, which is not surprising. And that cold temperature suppressed the blooming and also killed most of the flowers that had been blooming. Then after that, we had a very warm February, and the warm temperature carried over till the beginning of March, and the continuous warm weather pushed most of the cultivars into full bloom, and by the end of March and after mid-March, most of the cultivars have berries on them. So that's when the disaster happened, and on March 20th, around those days, and very cold, not too cold, but around 28 Fahrenheit degrees, cold weather happened, and that pretty much killed everything that already have berries on. So those are some pictures from a farm, Ferguson Farm in Alabama. So basically, everything that has berries on when the frost happened had pretty much no yield that year. You can see very scarce berries, and we were able to harvest However, we did have some selections from North Carolina which were about to bloom at the time of the frost happened or in the middle of blooming. And those selections were able to produce pretty good yield for first year plants. And so to name some of those, NC5289 is a really late blooming cultivar and endure the frost pretty well. And you can see the contrast of pictures there. And then NC3104, which is named Rowan now, it is also a late blooming cultivar and performed pretty well in that year with a late spring frost. So then going into 2024, we were able to evaluate more selections from Florida and Mississippi. And so remember that 2024, we did not have late spring frost, which is very unusual for us. But it does allow us to evaluate more cultivars and with a better weather condition. So this is a chart of progression of blooming date. We can see that some early ones are from Florida, some mostly from Florida, and some from Georgia and North Carolina and Mississippi, and they reach their 50% bloom date before February or in the beginning of February. And then after that, we have some early season cultivars, and mostly from Florida and some from Georgia as well. So they reached the 50% bloom date a little later than those super early cultivars, but also like still before March. And you can see like Patricia, Optimus, Colossus, and Sentinel. Those are some of the early cultivars for us. And then move on to the later season cultivars. Most of those are from Mississippi, some North Carolina, and we also have some named cultivars from Florida as well. So those selections, they bloom like in the beginning of March. So in a regular year, this would be good for Alabama without frost protection. Then again, we went to later season, and NC5314 or Rowan and NC5289 remain to be the latest blooming southern high bush that we were able to observe. And then we also have MS2162 served as another late variety for us. Since we did not have spring frost last year, and we were able to have pretty good yield all across the different types of selections. So if we start from the late blooming one, Rowan and 5289 continue to produce very well on yield. And then we move on to the mid-season selections, and NC5315 and New Hanover, pretty good. And for 1-year-old plants, Gumbo, Legacy, and Farthing had pretty good yield as well. And then if we move down to the early season selections, a lot of the Georgia collections, TH2929 and 3023 are those ones who stood out for their yield. So, and the younger plants like from Florida, like Falcon, Arcadia, and Sentinel performed pretty well last year in terms of yield. And in terms of fruit size, and the Florida selections had the largest berries, and the Georgia ones had very large berries as well. So that is an unusual year for us. So in general, the ones that are mid-season or late-season high bush cultivars would be more reliable for us. And another uniqueness of the super late-season selections is that they bloom super late and sometimes later than rabbit eye. So they are more frost tolerant in like very late spring frost events. And in the meantime, they also produce earlier than the regular rabbit-eye cultivars. So that will allow our growers to have better alternatives compared to traditional rabbit-eye cultivars. But if you're a grower like in Georgia and Florida and you have frost covers and frost protection, and those early season and selections would be pretty good options for you to target the early market. So some general impression about selection from different programs. For Florida cultivars, they tend to have like exceptional fruit quality. Fruit size is amazing, texture and taste are amazing. But in general, they tend to bloom a little bit for Alabama. And then for Georgia collections, very similarly, they have very good performance, good fruit quality and good size and amazing plants. And those Georgia plants are the toughest plants that we tested. They tend to have better survival rates and very strong root systems, very tough. However, like they also bloom a bit early for us, but might be good options for South Georgia and Florida. And for NC selections, and some of those selections are late blooming, which are very ideal to us. And they tend to have unique aroma and very good taste as well. The berry size is on the medium, during the medium range, and but overall fruit quality is really good compared to traditional rabbit-eye cultivars. And for Mississippi selections, and we also found some late blooming ones, and which will work better for us. They also have pretty good fruit size, but the fruit quality, like the taste, could vary between cultivars, and it's worth further investigation. Okay, so before I end the talk, can I also briefly introduce you a new smartphone app we developed for yield prediction? So this was done in collaboration with some Dr. Bell from University of Delaware and his PhD student, Pran Stein. So the idea is we want to use image-based analysis and machine learning models to automatically detect blueberries from a yield, a field image. And so we were able to collect 2 seasons of data and to train the machine learning model with 90 plants and 17 genotypes. Most of the cultivars we use for training are the southern high-bush cultivars commonly grown in Georgia and Florida, including like Farthing, Jewel, Star, and Meadowlark. And so with that information, we were able to train like models to automatically detect ripe and unripe berries from an image. So those images were taken just on a smartphone from the field. And so the model performed pretty well in terms of berry detection. And the accuracy is very high, and R-square score is about like 0.88. And then once we have the berry counts and we're trying to predict yield based on that, if we consider the average berry weight information that you manually measured, and then we consider the cultivar information, yield prediction is pretty high. But if we only use the general model and consider average berry weight, without knowing the genotype, the accuracy is slightly lower. So with that model, we were able to develop this smartphone app in the Google Play Store. And right now it's available on Android phones. And if you search for Blueberry Yield Analyzer, and you can test and see how it works for you. So the most reliable prediction would be the berry count. But you also have the option to go forward to estimate the overall yield based on the general model. So with that being said, like I want to acknowledge all the breeders who share their materials with us and my team members, and also funding agencies to make this research available. Thank you.
We have time for one question.
Oh, the requirement of cold hour, like I have a image here. So in a regular year, yeah, let's see if we can go there. Sorry. So in a regular year for South Alabama, it's about like 700 hours by the end of February, and for Central Alabama, it's about like 960 hours by the end of February. So if like our data in South Alabama would be more related to Alma, Georgia, but like slightly colder there. Yep. Thank you.
Thank you so much, Dr. Ru. And now we can continue with the next speaker, Miss Jessica Spencer from the North Carolina State University. So please, the floor is yours.
Thank you very much. My name is Jessica Spencer. I am a research scholar at North Carolina State University. Today I'm going to be talking about our blueberry program and how we build better blueberries for the South. First, I'll give a little overview of our breeding program at North Carolina State University, and then go over some of the research that is involved with that. And then I'll share with you about our recent upcoming cultivar releases. So NC State is in Raleigh, and that is where my colleague Hudson Ashrafi has his offices and labs, and he heads our program and does the majority of the molecular and genomic breeding. However, that's about an hour and a half from where our blueberry growers are located in southeastern North Carolina. I, however, am at the Castlehane Research Station, just a little closer by, near Wilmington. And we also have a 50-acre research site where we grow all of our blueberries. And so the soil is a little more similar to what you would find at the grower sites. And I'm also a little closer so I can go and kind of see them a little more when I get a chance. So we're breeding mainly for North Carolina suited cultivars, so that means 600 to 800 chill hours, acidic soil, and then our growers really like machine harvestable blueberries, so a lot of our focus has been on machine harvest. And then recently we're also looking at climate resiliency traits such as fruit splitting and self-fertility. So all these rain events, they create, cracks in blueberries, or the pollinators can't fly, and so these are important traits that we want to screen for. So it's a pretty lengthy process. As far as I figure, the least amount of time we can do it in is probably around 16 years, from the time that crosses are made to a cultivar is released. And it's a numbers game, so you have every year we try to look at around 8,000 seedlings and make about 100 initial selections. Then we grow those out and take another look at them and then advance those selections, put those in more trials and eventually with growers, and then hopefully patent and release. So my part in this is, the research part is to also optimize, I should say there's probably about 3 parts. The first is to kind of optimize all parts of growing blueberries. So I'm always doing these little kind of mini experiments, like germinating seed, trying to find a better way to germinate seed evenly, propagating, make sure we can propagate things and that they survive and root well so that we can have something to trial. Optimizing our field space. Seedlings take up a lot of space. If we can kind of condense that somehow, this is a high density plot I tried that's kind of similar to what University of Florida does. Doesn't work that great for us, but then again, I don't know, it's new to us too, so it's not something that we have done regularly. Next, I'm also doing little projects to find tools to screen for the traits that we need. So as I mentioned, the fruit splitting and cracking. We started submerging our berries in distilled water and kind of see whether or not they'll crack. Because we don't always get rain every season. So we want to make sure that on those seasons that we don't get rain, we can still have a method of screening our cultivars to know like what's going to crack and what's not, so we can try to not breed with those that are going to crack. We also have a machine harvester that we've modified to go over rows. So when we're doing, we want to test machine harvestable blueberries with an actual machine, but we have very small plots, so we've actually modified a harvester to be able to, this is an older kind, it's called a sway arm, but we've modified it so it can open the arms and kind of pass over the plots that we don't want to harvest and then harvest the ones that we do want to harvest. And then finally, texture and firmness, you've already heard a lot about that. We've been using the FirmTech for the past 20 years, now doing more with texture and the texture analyzer. And it's a great system, and as you've seen, the VacCAP program has found some really great parameters to look at the texture analyzer, or the texture, and kind of correlate that to important fruit traits. However, NC State has 2 of these, but they're 2 to 3 hours away from our location where we grow the blueberries. And also it's a really slow machine, like 2 to 3 minutes to run like a 10-berry sample. So we looked into finding something similar, and we did find something called the Agrosta Angeli texture analyzer. It's a lot faster. It kind of uses the plate to kind of move berries around. It's a lot cheaper, and it's fairly point and click, easy to use. We don't, however, have like it doesn't give us the data points because, VacCAP has all this nice macro that they made for the taxed texture analyzer. But we'll need to do that so that we can kind of get the same data. Right now we just get kind of raw data points that we'll have to try to translate into those curves. And then finally, another thing I work on is kind of streamlining the selection process. So we started using phone apps to take notes in the field and take data. In this case, Field Book by Trevor Reif, who's at Clemson. We were also starting a database using BreedBase, which is created by Lucas Mueller at the Boyce Thompson Institute. And we're also, as well, working on AI and machine learning. So in this case, we have a computer science PhD student named Jeremy Park. He's been using AI and machine learning to take all these pictures of blueberries we've been taking and get it to identify the berries and measure them and kind of spit out that kind of data. And it's fairly accurate and a lot quicker than having people try and hand measure or getting from just a few berries on a machine. We also have some goals of also categorizing, like, if there's any ripe or unripe ones in the picture, if they're shriveled or diseased. Hopefully measuring the color at some point and the stem scar size, and also identifying stem tears or any diseases. All right, so now we get to the fun part, our recent and upcoming releases, and recent is in quotations, because as you'll see, these are not all that recent. So in 2013, we released Pinnacle. It's a solid plant. It hasn't gotten much traction, but it's always worth trying in different locations, as you saw with Sushan's data. Some works better in some places than others. Our other is Heintuga from 2014. It's a pentaploid, so this one's really interesting because it is a cross from a southern highbush and a rabbit-eye. And so it's essentially seedless. It does have some pollination issues, but I think it might be a good candidate for using ProGib, which I don't think North Carolina growers use much of, but I think they do in Georgia. But I put a couple of plants in my greenhouse, and so it doesn't have any pollinators, and I sprayed it with some gibberellic acid, and it produced a whole lot of fruit. So that might be something interesting to look at. Next, we have NC 3104, or Rowan. I don't know how exactly you want to say that. It's actually an older cultivar. It was created kind of the same time as New Hanover, but New Hanover was released instead of it, just because it had a little more yield. But it's very similar, and we ended up learning that New Hanover, which was released in 2000, actually has a lot of stem tears. And so we've been working on releasing this so that as a kind of replacement for New Hanover, because a lot of our growers really liked New Hanover, but they just couldn't take it because of the stem tears. Next one, Dr. Ballington, he was our blueberry breeder who, quote unquote, retired in 2015. But he's also still doing a little breeding here and there, mostly on ornamentals and working with proven winners, if that's of interest to anybody, which I think it's really pretty, so. Finally, okay, these last two we are patenting this year. The first one is NC 5288. It's very early, very low chill. I'm thinking I'd really like to see it in southern Georgia or northern Florida just to see how it does. It's got a very large berry, really good flavor, very firm, nice upright architecture that might make it amenable to machine harvest. It does get redbacks if you harvest it a little too early. And in one location, we saw 5% tears. So there's might be some kind of mineral deficiency or something going on with that. And we haven't seen a huge yield, but that also might be because we've had frost damage in the past. So this is our yield at a grow location this past year. And I'll kind of explain how we do peak harvest weeks and main harvest. So I kind of average if the first week that we harvest something, that's week 1. So that this way you kind of get a general idea. So NC 5288 at this location was the earliest thing that we harvested, and it pretty much peaked before the 3rd harvest. And it got 9 pounds per plant, which I think I did the calculations in that. I didn't keep it on my phone though. Which I think would be pretty good, especially for Florida. But for North Carolina, we're trying to see if it'll be kind of a star replacement, because star has been very popular as a very early cultivar in North Carolina, but it just gets a lot of frost damage. Oops. And here's just a few pictures of what it looks like. It's very, like I said, very upright, very vase-shaped. There's a picture of the spring and then the fall after it's been mowed. And then here's some pictures of it as kind of at different grow locations at different ages. So this is all the same year, but they were planted different years. So the first is kind of like a 1-year bush, and the second's probably about 2 or 3 years, and then the third picture in the center is about a 4-year-old bush, I think. And then next we have NC 5289. This is one that Sushan has that looks really good apparently in Auburn in Alabama. So that's kind of exciting for us. It's something we've had our eyes on a while. It's really pretty. It's got really good, interesting, complex flavor. Again, it's maybe not going, for North Carolina, it might not replace Legacy because Legacy is has such a high yield, but it looks like it might outcompete Legacy in some locations. And I think I heard that Georgia is also looking for a replacement for Legacy, and so this might fill that bill. And just to kind of show you where it is in our harvest window, it peaks, for 3 years that we had it, it peaked a week to half a week before Legacy. But then we have this data from Sushan and her program just this year from southern Alabama and central Alabama. And you can see it's actually after Legacy in South Alabama, and then before Legacy in central Alabama. And then for some reason, this was a new planting in a new field. H2 is in North Carolina. It was a little bit before, or a little bit after, like about the same time as Legacy. But we also had a couple of issues with harvest, and we didn't get a couple of harvests done. So that might be part of that number issue right there. So yeah, it's really pretty. Here again at a couple of grow locations, what it looks like, a very compact bush, not as tall or upright as 5288, but it's really great. So if you have any interest, or if you're interested in propagating or growing any of these, please fill out this form. There's also a link to more information on our cultivars at this form if you want to click on it, and I'll also have this at the end. I do, however, have a couple more to show y'all that we're going to be trialing with growers this year. One of them is NC5303. It's an early mid-season, and then NC5305, which is more mid-season. And we mostly trial in North Carolina or with public institutions just because of how where we get our grant money from, and that's kind of where we feel our loyalties lie. But if any other growers outside the state have interest in trialing these, or if a nursery does, we are very much open to sending y'all plants, letting y'all propagate them, up to 200 plants or so, or potentially be purchasing them from a contract supplier that we have. It's a little bit, we're still trying to hash out the details because of legal issues and patenting, but it could happen. So, but for trialing, a trialing agreement will have to be signed, and you can't propagate more than the specified amount, and you can't sell the plants or specifically market the fruit. So you can sell the fruit, but you can't be like, oh, these are, NC5303 or NC State berries or whatever. You just kind of have to mix them in with your general population if that's what you want to do with them. So with that, thank you all very much. Sorry, my voice is cracking here. Any questions? Yes, sir. I personally have not, because that is a lot of money to talk to consumers and do that kind of taste flavor profiling, but I know that Massimo and Patricio both have done that kind of work, and it does seem like the crispiness and the crunchiness is something that is very desirable for consumers at this point. Although I have had one temp intern who likes the softer ones, so there you go. It's not, there's an outlier right there. Yes, sir. Hey. It's just not, the yield's not there as compared to 5288. It's got the same kind of timing as far as bloom. It might even bloom a little bit earlier, and I think it tears more. We saw a lot more tears, so I think we're not going to move forward with it. Yeah, New Hanover always tears. We haven't figured out why or how. I think if we had someone dedicated to kind of like cultural or managing blueberries, we could kind of figure it out and figure a way around that. But yeah. That's a good question. We're still trying to figure out our machine picking methodology, honestly. So, what about you? What's the best machine picking one you've seen? Okay. Yeah. Yeah, farthing is kind of good all around from North Carolina. Don't tell Florida I said that. So, yes, sir. We do not. We are hoping to work with Juliet and exchange some germplasm and materials, and hopefully, yeah, y'all can take a look at it at some point, because that would be great. Okay. Great. I'm trying to. Is MBG the same as Michigan Blueberry? Okay, because they contacted us and were interested in testing them, but I think they were going to still be testing them in Michigan. I'm not sure if they would have been testing them in Georgia as well. But yeah, we're hoping to get some with them as well. All right, great. Thank y'all.
Thank you so much. Thank you so much, Ms. Spencer, again. And now, last but not least, we have the next speaker, Dr. Gerardo Nunes from the University of Florida. So please, the floor is yours.
All right, well, good morning, everybody. It is quite the day to be talking about sun today, but for the next few minutes, I'm going to have an opportunity to catch you up on some of the research that we have been doing in my laboratory at the University of Florida, looking at light quality and intensity in the blueberry canopy. And to talk about light is, of course, to talk about a really important resource for the plant. When we think about how light impacts the plant, the first thing that comes to mind for us is photosynthesis. Photosynthesis is how the plants are going to make carbohydrates. And that formula there may mean a lot or very little to everybody, but when we think about photosynthesis, what I invite you to think about is the resources that the plant is going to have for growth, defense, yield, reproduction, and of course the ever important fruit quality. So when we think about light, we're thinking about one of those inputs. Without light, there's none of this for the plant to happen. In the context of light, we think of light in two very different ways. The kind of light that affects the plant for photosynthesis usually takes the name photosynthetically active radiation, and we usually abbreviate that PAR. PAR light is necessary for photosynthesis, and that's going to be a really big element of the story here, because when we think about PAR light, we're thinking about the light that the plant can use in order to make these organic compounds that matter. The other way in which light is going to affect plants is going to be light quality, which refers to the different wavelengths, or said otherwise, the different colors of light. And here we're going to see specific responses that are going to matter in the context of blueberry production. The first one that I want to introduce today is far red light. So when we think about how us as humans perceive light, we can see red light. We see red colors after all. However, plants have photoreceptors that go beyond what our vision can take. That's the part of the wavelengths that we call the far red. Far red wavelengths are very important for plants because when the plant has lots of far red light that is incident on it, the plant is going to start stretching. It makes those very, very long internodes. And if you have a long internode, you have fewer nodes, fewer branches, fewer fruiting points. You see where I'm going with that. We also have blue light. Blue light is going to be extremely important for the plant because when the plant receives blue light, the plant opens the stomata. Those stomata need to be open for the plant to be able to exchange gases. That means the plant loses water, but also takes CO2, which is important for the plant photosynthesis. And finally, we have UV light. And UV light is important for us because UV light is related to the accumulation of anthocyanins, which are important for the development of color in the fruit and the avoidance of post-harvest issues, such as the green or red backs that we have heard about earlier today. So in that context, I want to talk about light, but usually when I talk about light in Florida, where I'm from, but also throughout the Southeast, we tend to think about light as a resource that is plentiful. And we are not wrong. There's a lot of light during our growing seasons. But as I'm going to show you over the next few slides, the shape of the plant and also the time of the year can actually affect how much light is available for our plants to actually do photosynthesis or any of the responses you see here. The first thing that I want to talk about is about saturation. We have not been able to run these kinds of curves for every single cultivar, but for every one of the cultivars that has been run in the literature, such as the ones you see here and the ones that we have done in my lab, we have seen that traditionally, when we think about the light intensity at which photosynthesis saturates, we're looking at the neighborhood of 800 micromoles per meter square per second. This means that light intensities below 800 micromoles lead on to lower photosynthesis, and that light intensities above 800 micromoles are not going to be beneficial for the plant. Now, it's important to keep in mind here that in the middle of the summer, in a cloudless day in Florida, you're going to get 2,000 micromoles per meter square per second. So it's quite easy to make the assumption that light is always abundant for the plant. But as we started studying more and more about the blueberry canopy, we're finding out that that's not so much the case. The reason for that is that leaves and canes within the plant canopy are going to create shadows. And those shadows, of course, end up making it so that the interior of the canopy has far less light. What we see here is results from two different cultivars. You have Farthing to your left and Optimus to the right. Farthing, one of the more established cultivars. Optimus, a very popular cultivar now in Florida that is expanding throughout our region. And what you see on the y-axis is the fraction of the total photosynthetically active radiation. At the top, one represents full sun, the amount of light that was incident at the top of the plant. And now, as you keep on moving down on the y-axis, you see less and less of that coming in. As you can see here, in February, when these plants were fully deciduous, we saw that at the top of the canopy, we were getting very close to full sun. But by 0.6 meters, which is going to be mid-canopy in most of our growing systems, we start seeing far less light. Now, let's move along the x-axis. As we go from February to March, you can see overall the light that is incident in the center of the canopy, much lower. As we move into April, this is exactly when we're fruiting, March and April in Florida. You will see that the center of the canopy has even lower light. By May, when our season has ended and our plants are just fully leaved, the interior of the canopy has less than 1/4 of the light intensity of full sun. Now, let's go back to the numbers. We said that a full sun day in Florida gives you 2,000 micromoles per meter squared per second. The interior of the canopy only gets about 25% of that, meaning that in the center of the canopy, in a very, very sunny day in Florida in May, you're going to have leaves that are not photosynthesizing at their maximum. This is a really big paradox that we've been thinking about, because it's not something that meets the eye. Believe me, when me, my team, and anybody who's a grower in the Southeast, when we're out there in May, we're wearing sun protection, we're wearing hats, we're wearing long sleeves, because there's a lot of light. It's not very intuitive to think that the center of the bushes we're working with are actually not getting enough light for photosynthesis. So we took it back to our data loggers, and we started taking a look at what this means for our plants. The red dotted line here represents those 800 micromoles, the point of saturation for photosynthesis. And I just took a random snippet of a few different days in 2023. As you can see, there's plenty of parts of the day when the leaves are not going to be getting enough light for photosynthesis. That's of course problematic, because it means that we are spending water, fertilizer, pest and disease control in keeping leaves that look healthy but are not working so much for our plants. We also see that in farming situations where we're using high tunnels, the plastic of the tunnel, no matter how new or how recently installed it may be, actually also reflects some of the light away from the tunnel. And at every step of the day, as you can see here, with data from March 1st of 2024, at every step of the day, if you are cultivating under a tunnel, you are going to have far lower light intensity. Therefore, it is not so simple as thinking there's a lot of light outside, thus the bush will have enough light for doing photosynthesis. As we started digging into this idea of light intensity, we were using the kinds of sensors that also allow us to measure light quality. And this allowed us to take a look at the red and far red ratios inside those bushes. Looking one more time at the Farthing and Optimus at two different depths, 1.2 meters to the top of the canopy and 0.6 meters to the center of the canopy, we took a look at the red to far red ratios. At the top of the canopy, those red to far red ratios are approximately 1, meaning that there isn't an enrichment in far red light. What do we see at the top of the canopy of a blueberry? We see very short internodes, and in those very short internodes, lots of leaves, lots of branching points, lots of fruiting points. That's what we want. Now, if you take a look at what happens in the middle of the canopy at 0.6 meters, here we see an enrichment in far red light. Since there is so much more far red light in the center, this is the part of the plant where we tend to see those very long internodes. This is where we see fewer branching points, fewer fruiting points. And in many of the most vigorous systems that we see in Florida, we actually tend to see that in the middle of the season, we develop canes that are really long internodes. That is because all of that new material is developing in an environment that is far red light enriched. But up to this point, we had only tracked two very important, admittedly, but only two cultivars. And if you know anything about Florida, is of course that we have a very vibrant breeding program and that we have a very fast turnover of varieties. So we wanted to get some information about some other varieties and get a sense for just what kind of diversity exists out there for blueberries. So my postdoc set out an experiment in collaboration with Patricio Munoz's team, and we took a look at 29 different southern high-bush blueberry cultivars. These plants are 5 years old, grown under commercial conditions. They are hedged, usually as it is commercial practices in Florida, at about 65 centimeters from the ground, and they're usually hedged in weeks 18, 19, or 20, depending on just where in the northern part of our state you are. We use three very basic measurements: plant height, which reflects the vertical distance between the ground and the tallest point in the plant; plant width, which we measured across and in the road and reflects the distance, the horizontal distance between the furthest points in the plant; and the base angle, which represents the angle between the two furthest canes, assuming that they would connect at or below ground in the bed. When we started taking a look at this, we used RGB photography to get images of the plants, and then light interception using an LI-110. When we started taking a look at these phenes, the first thing we noticed is that, like everything with blueberry, there is great diversity. If you look here at the red line, that represents the height at which these plants were pruned. And the return from the prune is extremely different from plant to plant. All the way to the right, you have a plant like Sentinel, Arcadian, Florida Rose that more than double their size in the Florida growing season. Our measurements were done in the month of October. At this point, we assume most of the growth has stopped. On the other hand, you have plants that are not going to grow so much in terms of their plant height. For example, you have a few of the breeding selections, and Patricia, Scintilla, and Emerald, that are only going to stay up to about 1.2 meters. The average plant in the entire plot grew to about 1.5 meters in height. Then we look at base angles, and the base angle is actually one of the most revealing parts for us. In Florida, we're generally planting blueberries 3 feet apart in the row, or a little bit less common, but we also see farms that are going to plant about 2.5 feet apart in the row. Now, our planting in the row usually assumes that the plant's going to have enough light and that it's not going to be competing with one another. Well, when we took a look at the base angles, we started to question whether there should be a one-size-fits-all approach to this. What we have here is that we encounter plants that had very wide base angles, all the way to the right-hand side, Windsor, Scintilla, and Optimus, but also plants that have very narrow base angles, such as Snow Chaser, Spring High, and Sweet Crisp. And I think the best way to explain this and to show it is to just put Optimus and Key Crisp side by side. Optimus has a very wide base angle, approximately 150 degrees between those two red lines in there. And that's quite different than what we see when we compare, for example, to Key Crisp, that has a much narrower angle, usually smaller than 90 degrees. Therefore, when we started looking at this, we started thinking, should we actually be planting both of these cultivars with the same spacing? The answer is probably not. So what we have done since is we have started an experiment looking at different planting spacings. Now, plant spacing research is research that has been done for decades, and it has been done decades even before I started studying plants. The approach we want to take here is we want to put these plants on the ground and take a plant modeling approach to be able to understand not just how to space Key Crisp and Optimus, but hopefully to build some baseline knowledge that's going to allow us to make a recommendation the next time Patricia has a cultivar with a very different base angle. When we looked at the canopy characteristics, we found that there were two important things that are closely related but not the same. One of them is the leafiness, which we measure here as the leaf area index. As before, we found lots of diversity. There's plants that make just lots and lots and lots of leaves, like Optimus, Colossus, and Wayne. But there's also plants that make canopies that are much more meager, much smaller, such as Patricia, Sweet Crisp, and Scintilla. When we look at these two extremes, we're looking at the number of leaves, but we're also finding that some plants make leaves and they have absolutely no problem shading one another. However, we have plants that make leaves that do not shade one another and essentially create light pockets. This is what we have measured as canopy density. Notable for us is that you can have a lot of leaves, but also a very not dense canopy. So we are looking at two very different ways of understanding this canopy. When we looked at canopy density, we were particularly enthused about the fact that when we consider the density of this canopy, we're looking at canopies that are less dense, that allow the light to penetrate through. After all, we started looking at plant shape because of this interesting light. So when you look at the lower canopy densities, for example, Prima Donna, Patricia, Scintilla, and Emerald, those canopy densities are lower, and they do, in fact, let more light through. Well, ultimately, of course, we had to go in and measure the light. So we measured the light intensity this time around at 15 centimeters from the soil. This is about as low as you could possibly measure this, considering the kind of sensor we use. And we saw once again that the plants that have lower canopy densities in fact allow more light through and likely have leaves in the inner canopy that are constantly working, constantly photosynthesizing, and of course we assume also going to have better outcomes. So in summary, from our first foray of research into canopy light and canopy shape in southern high-bush blueberry, we found that light intensity and quality changed throughout the blueberry canopy, and that this usually has no relation to the light that we perceive is happening everywhere else in the farm. This has us thinking about what exactly is the right light sensor that we should be using. Most of us probably have in the farm a pyranometer or a pyrometer in the weather station, and that's only one of the important points of information that we need to consider as we try to understand better and better how to optimize cultivation. We also found that there are several different canopy architecture types in southern high-bush blueberry, and that among the 29 different cultivars that we have studied, they actually group relatively neatly into several categories. Finally, we found that canopy architecture impacts photosynthetic light interception, and anything that impacts photosynthesis is going to impact yield and other agronomic outputs. So what we're getting busy on as our season for these kinds of measurements actually starts at the end of January is we're going to start investigating the role of light interception on fruit ripening. Of course, one big overarching goal for our industry is to be able to mechanize seamlessly. And to do that, we need to have all the fruit become ripe at relatively the same time. So we're trying to concentrate ripening, and we believe that light could have a role in that. We're also going to be investigating the year-to-year stability of the plant architecture traits so that we don't have to make multiple measurements on the same plant over and over. And we actually have the ability to make some predictions about what farming systems are going to work better than others. I will wrap up by acknowledging that the majority of the data that I presented today was collected by my graduate student Martin Sapien and my postdoc Ronnie Kader. We've done every bit of this work with funding and collaboration from Patricia Munoz's program at the University of Florida. My contact information is in the screen, and with that, I'll be happy to take any questions. Yes, so it depends. If you're working with some of the very, very tropical southern high bush, you can get great photosynthesis up into the 90s. But as we move northern, so if you're working with the southern high bush that are much more from the continent and not from the Florida Peninsula, you start seeing that photosynthesis starts going down at about 90 degrees Fahrenheit. And then if you're working with a northern high bush, those usually start dropping their photosynthetic rate in about the high 80s. Sure.
What light level the leaves start to fall off as far as, in other words, could you go in and say at 200, put a light probe in there and say, if you do a hand pruning, it's getting too shady, at some point the leaf is going to get parasitic, they're able to fall off.
So we've studied the leaf senescence in the context of evergreening. So we've never looked at what light intensity is necessary to keep the leaf viable. What we have found when we looked at evergreening and retaining leaves through the winter is that blueberries are exceptionally good at holding onto the leaves, and that seems to be much more associated to the nitrogen fertilizer than to the environmental conditions. So we had an opportunity to set an experiment with plants inside a tunnel and outside a tunnel, and with different rates of nitrogen nutrition through the winter. The nitrogen led us to holding onto the leaves. The air temperature led us to having high photosynthetic rates. So for northern Florida and throughout the south, our way to achieve that earliest production is always in tunnels because they combine both. But we've seen the blueberries hold on to their leaves sometimes to a fault. What we have seen is there's a big variety in the kinds of plastics you can get. If you are buying at the top of the market, you're going to get a plastic that usually is very UV stable, which for the purposes of farming, it means longevity, right? So if you get a plastic that's UV stabilized, you're going to get, in Florida at least, you're going to get 3-4 years out of your plastic. If you get a plastic that's not UV stabilized, it's a 1-year plastic. So that's your trade-off. There are plastics that are traded with the idea that they are going to be neutral to the spectrum. So the same spectrum of light you get from the sun will make it through the plastic. That is not particularly true in my experience with all plastics. There are plastics that in fact are going to give you enrichments, particularly in the blue part of the spectrum. So we haven't seen a whole lot of plastics that are going to lead on to changes that will give you more far red light. So we haven't seen major architecture changes on the plant because of the far red. So at the very top of the canopy, you have the leaves that are providing for most of the plant. The leaves in the lower part of the canopy generally only provide for themselves. And the leaves in the interior, those are, using Gerard's words, those are parasitic. The leaves in the interior of the canopy could photosynthesize. They just don't get light. So at the top of the canopy, that's where the leaves that are doing the work are for us. Thanks. Right.
I'm not allowing my plant to go, why then do you expect any different from activity or?
So one of the things that we want to do next is to actually go back and measure this in a plant that is trained to have the narrow base and to open up at the top. Because what I want to make sure is that I have a better idea of what happens in those leaves that are ultimately lower, but also horizontal, right? Because the leaf orientation is also going to change. But that's not something that we have done to date. Not necessarily. To our surprise, every variety we have measured photosynthesis on, and my lab does a lot of photosynthesis work at this point, so we've measured some like 35 varieties. There doesn't seem to be a direct relationship between the growth rate of the plant and the photosynthetic rate. That's usually how I think of bigger. Then it comes back. Mm-hmm. That I think we think about it the same way. We might be using different words for it.
Last question.
If you want to give any work with bushes that are pruned at 3 to 4 feet, because the bushes, I think 65, 70 is, that's only 2 feet, right?
About 2.5.
Cutting
Yeah.
out all that brushwood, that's a really hard cut. Most of the commercial farms will start out somewhere between 36 and 40 inches, going up several inches a year. So that's
Yep.
really interesting to look at some bigger bushes, see what's happening with the light.
This was admittedly a sampling of convenience. Patricia had been growing these plants for quite some time, and we wanted to be able to land this idea on a broad genetic basis. We are following up with these ideas in a couple of different directions, and we will make sure to keep this in mind. Thank you.
Thank you again, Dr. Nunes. Unfortunately, that's all the time that we have for today, but I would like to thank you again, all of our speakers, and for the time and the expertise that they show us today. And to all attendees, thanks again for attending this presentation, and have a great day.